Welcome to Planet MicroCap. I'm your host, Robert Kraft, and joining me right now is Jeffrey Meckler. He is the CEO of Indaptus Therapeutics. It's a publicly traded company. The symbol is INDP on NASDAQ. And Indaptus Therapeutics will be presenting at our upcoming investor conference, the Planet MicroCap Showcase, taking place in Las Vegas, April 25 through 27, 2023. For more information, to meet with Jeffrey, to see the Indaptus presentation, please visit planetmicrocapshowcase.com. And with that, Jeffrey, thank you so much for joining me today. How you doing?
Hey, thank you, Bob, for having me.
It's great to have you. So this is actually our first time doing an interview together. So can you start us off with that quick overview and history of the company?
Sure. So Indaptus Therapeutics was actually created a little over a year and a half ago, and it was formed through a reverse merger. The technology was really developed over the last 8 years by our chief science officer. His name is Dr. Michael Newman. He's the inventor of the concept and the technology. And so what we did was we formed this about 18 months ago. We raised about $30 million in a PIPE. And our goal was at that point was to finish out all the work necessarily to file an IND, which is a drug registration for a new drug. We did that last year. We got clearance from the FDA to start our trial. And then we initiated the trial right at the end of the year, and we've dosed our first patient February. So the trial has started and we're underway. So it's been very exciting times and a lot of progress so far.
Very good. So Jeffrey, what would you say makes the company unique and different compared to some of your peers out there also working in a similar space?
Yeah, so our company, the goal is simple, is to really engage the immune system in a way to cure disease. And we feel that to have effective and efficient activation of the immune system, you need to both utilize the innate and the adaptive immune system. And I don't want to get too technical here because I know our audience are not usually a bunch of doctors, but really the concept dates back hundreds and even a couple thousand years to the observation that people in the presence of an infection would shrink and see complete remission of cancer. And so in the late 1800s, a doctor actually here in New York City, his name was William Coley, came up with the idea of giving killed bacteria to patients as a way to elicit that kind of response. So kind of getting the body to act like there's an infection and go after the cancer. And in some ways, there was a lot of success. There were lots of patients who had their tumors resolved through the Coley therapeutic. The problem was we didn't really understand how this worked, and it was still very toxic. So what Dr. Newman did was going back into the drawing board and saying, look, modern approaches are looking at specific targets. So if you look at almost all immuno-oncology therapies, they're looking for the silver bullet, that one little switch that if we turn that enzyme on or turn that agonist on, that we will cure cancer. The reality is biology is a lot more complex. The systems of our innate and adaptive immune cellular pathways, it doesn't work that way. And this is why the best-selling drug, the #1 cancer drug in the world, only works in about 1 out of 5 patients. The reason is it's more complex than just turning a single cellular pathway on and off. So what Dr. Newman came up with the idea of, what if we could engineer bacteria in a way that we could reduce toxicity? And the analogy I like to say is when bacteria have all of these agonists on it, and what we're doing with the Decoy technology is we're in the mixing room of a recording studio, and we're turning down and turning up the volume of different channels. And the first therapeutic is we're turning down that channel. It's the lipopolysaccharides, the LPS4, which is the most potent immune stimulator. By turning that down, we make it safer. And then the rest of the immune profile can be received by the body, the immune system activate and do what the immune system's designed to do, take foreign non-processing cells out of the body. So I hope that was a easy enough way to explain it.
No, absolutely. Listen, when it comes to biotech interviews, I love analogies. That's for sure. And my audience does too. But I want to talk actually about some recent news. I mean, one of the reasons we're speaking today is that the company just put out some data results from your, correct me if I'm wrong, your preclinical studies that came out. Tell us a little bit more and why this is so significant for the company.
Yeah, so this week we were at AACR, which is a American Association of Cancer Researchers, and we were able to present some new data on preclinical models exactly. But what we're able to do is do two big things at AACR. One was it's the first time we showed product characterization. So what we do is we get this bacteria, we kill it, we process it in a way, but we had never actually shown all the different toll-like receptors and different nod and sting, all the different agonists. So we actually were able to show profiling data of this combination activity. The second thing we were able to do that was really novel was for the first time we showed tumor eradication data as a monotherapy, single agent. So what we did in an antigen presenting model, which is a HER2 breast cancer model, we were able to give our therapy, and 2 out of the 5 mice in this model had complete regression. So just by giving Decoy, they were able to have, and it was a durable response, have their tumors resolve completely. Now, what we have been able to show prior, and we showed it again here at AACR, was in combination with multiple different therapies, we're synergistic, and we're able to show 100% total tumor eradication in several models where we combine it with anti-PD-1, which is the #1 selling cancer agent. So that was the big highlight. We had a bunch of other data that we were able to show in our session, but those were the two new things that were big and exciting.
Absolutely. So, I want to keep this relatively, we got 5 to 10 minutes here. So my next question then for you, or it's a two-part. So, I mean, this clearly is probably some news that you were hoping to put out this year as some sort of catalyst for the company. So I'd love to, from you, in your opinion, from what you can tell us, what would you say are some of the other value drivers for 2023? And also, how does that tie into maybe regarding some of the reasons why folks might want to meet with you in Las Vegas?
Sure. So 2023 for us is the year that we really start generating human data. So I think the biggest set of catalysts, and we have a chart on our website as well as on our presentation that shows that this year we're doing the ascending dose clinical trial. So I think the big series of catalysts is as we complete cohorts, which is, we're starting at a certain dose and we inch up to be able to read out on the safety of the program. And like I said, we've initiated that. We'll be throughout the year updating on, we completed this cohort, this is what we saw, this is what we learned, this is what we're doing in the next cohort, all of that type of information. And that's really exciting. Our goal is by the end of the year to get to our multi-dosing part of our study. We think that's the area that we'll start seeing signals in what tumors respond better, what settings work better. In addition, we're working on the platform in a number of really interesting ways. And I think later on in the year, we'll be able to talk more about how we're learning, as I use that mixing table analogy, how we're learning to construct that a little bit. In addition, I think we have some studies that are ongoing in other areas where we're looking to see how best to use this. This is not just a cancer agent. We actually have data in hepatitis B, a virus that has no cure. And we show activity that in a number of models that the current state-of-the-art, standard of care doesn't. And so we think there's opportunity in a combination therapy to prime the immune system and let these antivirals work better. And we have data in HIV and HBV. I think we're going to do some more work this year on the virus front and see if we can generate some more data. So I think the catalysts for this year are getting through the different cohorts in the single ascending dose study, starting the multi ascending dose study, expanding the portfolio, and then looking at different opportunities in different viruses and other ways to utilize this platform.
Very good. All right, Jeffrey, I think we're there. So with that, where can our audience go and find more information on Indaptus?
So just visit us at our website. It's www.indaptusrx.com. So INDAPTUSRX.com. And we have all the information up there, and we'll be looking forward to hopefully seeing some of the people that are listening right now in Las Vegas.
Very good. Well, Jeffrey, thank you so much for joining me today. Really do appreciate it. Good luck. Stay safe, and I'll see you in Vegas.
Thank you. See you then.